Thank you very much, Robert, and it's always a pleasure for me to be here because it dates for many, many years. I think we are collaborating for at least 2 decades together with you and with people from your laboratory, and I'm very happy to be here and to present this lecture for a cream of the meeting, because there's only people who stay at the till the very, very end. So I'm going to talk about. Can we do the full screen, please? I'm going to talk about astrocytes, astroglia in aging and neurodegeneration, but of course I want to start with what Robert already has mentioned, that this is a war now waging in Europe, first after 1945, and I really pray for people in the army of Ukraine who are fighting against barbarian horde, and I hope that this new Nazi country is going to be totally destroyed in the process. So we are coming back, astroglia in aging and neurodegeneration, and I'm going to talk about certain debatary issues today, in particularly challenge the idea of the so-called inflammaging of the brain, which is very popular because somehow this idea of neuroinflammation become almost toxic around neuroscience, and there were about 60,000 papers published last year which use this particular word, neuroinflammation. And I will try to show you today that in most of the cases this word is misused and probably abused, and aging brain is not inflamed brain, as many people try to make us believe. So the brain, of course, is an incredibly complex machinery which is made from several cell types. Of course, there are supportive cell types like brain vasculature cells, smooth muscle, endothelial cells, and so on and so forth. And there are about 200 billion of neural cells, which include neurons, of course, which is about half of them. About 100 billion of cells in our brain are neurons, and those neurons are chiefly responsible for information processing, for sending signals, for receiving signals from the periphery, for transforming them, for analyzing them, and then sending signals back to the muscular system in order to produce behavior. And the second half of the brain is, of course, neuroglia, which are supportive cells of the brain. But we have to admit that we have to really be very careful when we are talking about the brain tissue, because all these cells are born together, they work together, they die together, they undergo disease changes together, and they cannot survive without each other. So, in a sense, this is what is called brain active milieu, is a continuous communication between those two big words, the word of electrically excitable neurons, the word of electrically non-excitable glial cells, which are doing everything to support these neurons. And that is a result of evolution of the brain, which started about 500 million years ago from a very simple diffuse nervous system, and then, of course, it came to vertebrates. And what happens in evolution is that the cells were separated with their functions. So neurons are responsible for signal transmission and signal processes, but they cannot do anything in order to support them, in order to survive or protect. And all these supportive functions, homeostasis of the brain, and protection of the neurons went to neuroglia, and they are indeed the ultimate supportive cells which are making brain what it is. Brain aging, and that is, of course, a general understanding that aging is associated with decrease in cognitive ability. And as Shakespeare said, what is about old people? They have a plentiful lack of wit, together, of course, with most weak hams. So that is indeed somehow a prevailing idea of that. But now, if we start to look more carefully into different organs and systems, we would realize that the brain, the nervous system, the central nervous system, actually resists aging much better than any other organ and system. And if, for example, you will put a 50-year-old professor running 100 meters together with a young student, obviously the student will win. Because over these 25 or 30 years which separates them, muscular system, cardiovascular system, everything went through a very substantial decline. At the same time, when we are talking about cognitive abilities, at about 50, 60, this is a summit of development and of functional capability of the nervous system. And why is that? It's because, of course, of a plasticity of the brain. So over our life, we are learning, and this learning changes the brain and keeps it at a much better stage than other organs and systems, providing there is no disease. Obviously, when disease comes, when pathology comes, all this intricate machinery is destroyed, and then clearly we have cognitive abilities. And this particular notion of the brain being the most resilient towards aging is very old. And here is a book which was published, as you can see here, in 1889. And this book is covering results of a long-lasting, about 10 years of investigations of old people in England. So altogether, they looked for about 900 people aged 80 or more, and 74 out of them were centenarians. And again, that is the conclusion to which they came, that the brain in many held out as well or better than other organs. And when they looked in centenarians, for example, there were only 2 out of 74 with memory problems. And altogether, memory was good in more than almost 80% of people in their study. Now, how to interpret it is very difficult because many things has changed, and many different interpretations are possible, but nonetheless, it gives some hope that indeed something has happened probably over the last 100 or so years, which increased the propagation of dementia and which led to what some people call epidemic of dementia in our world. And if something has changed, we can identify it, we can remove it, and then, of course, we have a hope to cure it. And today I'm going to talk a little bit about inflammaging, and that particular concept was introduced by Claudio Franceschi and his co-authors about 23 years ago, and they looked at the whole organism, and they found certain similarities between inflammatory responses and performance of the organism, and so they start to look into aging as a chronic inflammation. And of course, this particular theory was applied to the brain, and now it's quite popular in neuroscience that brain is chronically inflamed, and in the aged brain we have a chronic inflammation which actively destroys the brain. So let's look into it, but first of all we have to make a digression and to realize what is neuroinflammation. And of course, inflammation as such is a stereotypic response of any tissue to trauma of whatever genesis. And that was described 2,000 years ago by Aulus Celsus, who made the first 4 major pillars of inflammation, the major symptoms, which are, of course, tumor, rubor, color, and dolor. And some years later, Claudius Galenus added the 5th one, pillar of inflammation, which is loss of function. And neuroinflammation as such is also very old, so the first written account for inflammation of the brain, which was called phrenitis, you can find in writings of Posidonius of Byzantium of about 4th century AD. So, what is inflammation? What is neuroinflammation? And in principle, all these major symptoms of inflammation, they have their cellular and molecular correlates. And those are release of pro-inflammatory cytokines, activation of macrophages, recruitment of leukocytes, and local tissue damage. All this is a result of the invasion, of the damage of the tissue. And inflammation is a process by which circulating leukocytes and plasma proteins are brought to sites of infections in tissues, and that is more or less a general definition of inflammation as such, which you can find in any decent textbooks. And now I would like to honor Elena Galea, who unfortunately died this year. She was a good friend and very important gliologist, and they published her last paper was published in 2023, just a couple of weeks before her death, in which she and Manuel Graber are actually looking into this neuroinflammation and make a very nice critical analysis. So, what was neuroinflammation? For a long period of time, neuro was simply topical. So there was no difference between normal inflammation or inflammation of nervous system. It just simply happened to be there, and therefore this neuro was there. And what was neuroinflammation? It was inflammation of the nervous tissue triggered by breach of the blood-brain barrier. So by a direct damage to the brain, either mechanical or biological, or chemical. And inside neuroinflammations are very exactly the same cellular and molecular hallmarks, like release of cytokines, reactive gliosis, which is corresponding to activation of tissue macrophages in other tissues, infiltration of monocytes, cell death, and local tissue damage. And then about probably 25 years ago, a logical break occurred in which identifying single hallmark of inflammatory process becomes sufficient to consider the process inflammatory. And mostly that was what we call reactive gliosis. So when people were seeing brain tissue with some increase in staining for astrocytes or increase in staining for microglia, they thought that this is reactive cells, and that signals neuroinflammation. And that is, of course, a logical break, which leads to a conceptual deadlock. And that is very important because, indeed, if the process is inflammatory, then we can cure it by using anti-inflammatory drugs. Now, I asked psychiatrists, it was a wonderful session before, and there are, of course, lots of papers which say depression is a neuroinflammation. Can we cure depression with ibuprofen? Probably not. The same applies to all other diseases. And I think it's really very important because this conceptual misinterpretation actually may and does have a therapeutic consequences because obviously we cannot cure most of the neurological diseases by using anti-inflammatory drugs. So in a sense, there are classical neuroinflammatory pathologies like brain trauma, infection, stroke, and autoimmune attack. And all other pathologies are not initially inflammatory at all. So inflammatory component may come when we do have a breach of blood-brain barrier, which does happen in certain diseases, for example, in diabetes. But before that, there is no inflammatory component whatsoever, despite the presence of cytokines and despite the presence of reactive glial cells. An aging brain, the general view of aging brain regards it as chronically inflamed with a prevalence of reactive glial phenotypes. And today I'm going to guide you through astrocytes, which are the main homeostatic and defensive cells of the brain, and to show you what happens to astrocytes in aging. So astrocyte, the term is, astrocyte of course means star-like cell. And from where it all comes, it comes mostly from studies of Camillo Golgi, who introduced a black stain reaction back then in 1872. And he was able to stain many different cells of the brain, and in particular, he stained parenchymal glia. And when this parenchymal glia is stained by Golgi technique, it looks indeed like stars with radial processes. And Golgi even realized that many of those cells, almost all of them in cortex, for example, they do send processes to the blood vessels and make these structures which are called end feet. And Golgi was the first to say that this is an interface between the blood and the brain parenchyma, and indeed there is a very important part of neurogliovascular unit which connects circulation to the brain parenchyma. The term as such was introduced by Michael von Linosek, who at those days was working in Bern, but he had a sort of a prophetic soul, because actually that's what he wrote. I would suggest that all supporting cells be named spongiocytes, and only some of them be named astrocytes. And indeed, now we know with advances of imaging technology that astrocytes do not look like stars at all. They look like sponges or bushes. And so, indeed, the name spongiocyte would be better, but the name of Astroglia, of course, is of common use. Astrocytes belong to a much larger class of the cells, which is called Astroglia. And these astroglial cells are cells of neural origin, so they are scions of radial glia, which is a major type of glia in embryonic brain. And they include many types of parenchymal astrocytes, and only some are shown here, so this is really very heterogeneous population of cells. They include many types of radial astrocytes, like Bergman glia and cerebellum, or Fananas glia. And what is interesting, the radial glia, which are neural stem cells, are also belonging to astroglia. So they are proper astrocytes with radial morphology. They have the same markers and astrocytes, the same physiology, the same function, plus on top of them, they are able to produce asymmetric division. glia of ventricles, which makes the walls of ventricles, ependyma glia, choroid plexus cells. They're also belonging to astroglia. They have the very similar physiology, they have the very similar calcium signaling, they have gap junctions and so on and so forth. Unfortunately, in the literature, very often they are called epithelial cells. They are not. They are the same science of radial glial cells or neuroepithelial cells, as all other astroglial cells in the brain. Importantly, astroglia is not the most numerous glial cell populations. They are about 20 to 40% of all glia, depending on the region. So please don't start your paper saying that astrocytes are the most numerous glial cells in the brain. Importantly, again, humans have several types of astroglial cells, which are absent in all other species, in particular interlaminar astrocytes, on which I'm going to say a couple of words later. What astroglial cells are doing? They are doing everything. They are supporting homeostasis of the brain at all levels of organization, from molecular homeostasis to cellular, metabolic, organ, systemic, and of course they are critical for defense and neuroprotection. How to study glia? It's not a trivial question. In particular, it's not that easy to visualize and characterize these cells because we don't have a universal marker. Glial fibrillary acidic protein, or GFAP, has been considered for a long time to be such a universal marker, which is true for experiments in vitro in culture, but in normal healthy brain, most of the cells, most of astroglial cells, do not express GFAP at the level of detection. So we need other markers like protein S100 beta or glutamine synthetase. And what is also important, GFAP reveals only a very small portion of morphology of astrocytes because it stains only cytoskeleton. And when we use more modern technique, like injection of lucifer yellow, for example, or expression of genetically targeted fluorescent dyes into astrocytes, you see this is real morphology of astrocyte. It really looks like a sponge. Why? Because it has very, very many small processes in the periphery which are covering synapses and giving this cell this spongiform appearance. Now, another digression, GFAP, is it an accurate marker of reactivity? And that is very important because in many papers, just increase in GFAP is interpreted as pathological response. So first of all, as I already mentioned, GFAP misrepresents morphology. So that is an old experiment made by Miloš Pekny. So this is GFAP staining, and then the cell was injected with lucifer yellow fluorescent dye. And now you see this is a true morphology, and only a very small part of this morphology is revealed by GFAP. Second, GFAP is an incredibly labile protein and can go up and down very rapidly in response to normal physiological stimulation. And that again led to many misinterpretations, like for example, in this paper, so this is control brain, and then those animals were put in a running wheel for some days, and a week later, we looked for the brain. Now you see many different stained cells, GFAP positive cells, and what was the conclusion? The conclusion was that they proliferated. Now in reality, we know that astrocytes are deeply postmitotic cells, they do not proliferate, so what happens, they upregulated expression of GFAP. And if they would wait for another week, all that would disappear. So it's a very labile protein. And it can change even during 24-hour cycles. So this is third ventricle. GFAP staining, you see over 24 hours it goes from a quite substantial labeling of astrocytes to almost none. And that is pure physiology. And again, if you put animals to physiological conditions, to physiological stimulations like physical exercise or enriched environment, or for example birds, some of them are migrating and some of them are sitting in their nests, you would see what happens. The astrocytes, they upregulate GFAP and they become much more complex. And that is pure physiology. So more or less we have to be very careful when interpreting staining with GFAP. Couple of words of pathology of astroglia. And as I said, evolution made a distinction between two different cell types. And when lesion comes, what happens? Neurons, they can't fight. What neurons do, they usually shut themselves down to limit ATP expenditure and to try to survive. Glial cells, in contrast, undergo an evolutionary conserved program which is called reactive gliosis. So they change their biochemistry, physiology, morphology, and they try to fight against lesions. They try to protect the brain. And if they work properly, then everything is fine. If they fail, then of course disease progresses and we have cognitive deficits or neuronal death and ultimate death of the brain. However, pathophysiology of astroglia is much more complex than only reactivity. As you can see here, this is more or less classification in making at that particular moment. And what we probably can distinguish between two axes. One is allostatic, that is changes in order to protect, changes in order to keep homeostasis. And at the very end of it, this is astroglial reactivity, which is again very complex. Could be proliferative, non-proliferative, and so on and so forth. Astrocytes are cells and they can die. And they do die in many diseases. And morphologically, it is seen in the form of plasmatodendrosis or fragmentation of processes. There is a big class of changes in astroglia, which is atrophy and loss of function. You can see this very long list of diseases. In particular, it's very important for psychiatric diseases. And now we start to identify a totally new class of astrocytopathies with aberrant astrocytes which drive the disease. An example could be Alexander disease, in which astrocytes express mutant GFAP, which results in vanishing of white matter, so we still have no idea about mechanisms. Because reactivity and astrogliosis are implicated for inflammation, of course, a couple of words about it. This is known for a long time, so this is a picture from 1927, which actually shows all the major characteristics of reactive astrogliosis in response to trauma. So, what do? I have to move? Yeah. In response to trauma. So here is a stab wound, and that stab wound will be then replaced. So what we have in this situation is that stromal elements go into the damaged cell, and then they make a fibrotic scar. And here are the changes in astrocytes. So distant to the trauma, astrocytes are still complex and physiological. Closer to the trauma, they start to move towards the trauma. They start to proliferate, which is very important. And they make a barrier. So, of course, you also read quite often words in the literature like glial scar. So glial scar doesn't exist. All scars are fibrotic in nature by definition. What astrocytes do, they make a barrier which delineates the wound from the rest of the tissue. And this barrier is absolutely important for closing the wound, for healing the wound, and for post-lesional regeneration. And if you block reactivity of astrocytes, you exacerbate the pathology. So for a long time, reactive astrogliosis was regarded by neurologists as a detrimental pathological response. In reality, it is not. It's a survivalistic response which helps the brain to limit the damage, and it helps the brain to regenerate. So more or less, exactly like we have in inflammation in the skin, inflammation of the skin, then the wound is closed, it's replaced by fibrotic scar, and the tissue is more or less functioning again. So astroglial reactivity or reactive astrogliosis, this so far can be used interchangeably, is an evolutionary conserved, primarily defensive response of astrocytes to pathology. And there are many different phenotypes, and all the trials to make a sort of a Manichaean classification and divide cells into good, bad, neurotoxic, neuroprotective, A1, A2, or for microglia, M1, M2. This is a real misinterpretation which really harms the field very much. In particular, I want to say a word about neurotoxic. So far we don't have any evidence demonstrating that astrocytes or microglia can produce de novo toxins which are killing the brain. In fact, the main toxin of the brain is released by neurons every day, and this is glutamate. So whenever we do have detrimental effects on neuronal survival, is not because the cells start to produce de novo toxin, but because they lose their function. And in particular, they cannot, for example, clear glutamate, and that signals the brain, the death of the brain. Aging brain. So first of all, a word about atrophy, which I mentioned. So this astroglial atrophy is observed in very many different pathologies, addiction, schizophrenia, depression, epilepsy, you name it. And that is also, as you will see, is observed in the aged brain. In principle, this particular slide shows you what was about aging brain for about 10 years ago. And as you can see here, immunocytochemistry can reveal anything. There are some areas in which cells become more GFAP positive, less GFAP positive, more S100 beta profiles, or less, or glutamine synthetase is exactly the same. So in different regions, it was total mess, and it was very difficult to understand what's going on. Now, if you start to use a modern technology, so those cells are now perfused with fluorescent indicator, that's what happens in mouse, young, adult, old. So during maturation, of course, astrocytes become larger because brain grows up and it needs more support. But then when it comes to old ages, then astrocytes shrink. And this is their territorial domains, and you see here they shrink really very substantially. And what is also very important, their complexity is going down. Now, what makes complexity of astrocytes? There are, of course, these primary processes and these myriads of very, very tiny processes which has about 100 nanometers in thickness, which don't have organelles and which cover synapses. And those processes actually go down quite substantially in old age. How to study them? It's very difficult because they are very thin. So for that, we use what is called volume fraction. So we measure fluorescence in the region of the astrocyte when these processes are, and that is like a gallo, and then divided by the fluorescence of the soma, assuming that soma has 100% of occupancy. And this is volume fraction. And that's what happens with volume fraction, so it's a fraction which is occupied by these very tiny processes. What happens in aging? It goes down very, very substantially. Physiology sort of matches it, and this is, for example, density of major receptors and glutamate transporter. You see they increase in early months of age, and then they fell down and stayed depressed at a low level in advanced ages. And that translates to functional deficiency, and that is, of course, potassium buffering and glutamate clearance. And potassium buffering in old age goes down, and glutamate clearance, these are current of glutamate transporters, is also going down. And that translates at the end into an impaired long-term potentiation. And now we can prove that this is astrocytic because we can take a normal, so this is adult animal, this is old animal, but if we take just an old animal and block astroglial glutamate transporter with a specific blocker, it will have the same effect as aging. So this is really astrocytic. Now what about humans? And that of course is very important because humans are different. And those are experiments from freshly isolated cells. This is access tissue for brain surgery of different ages. There are astrocytes, 34, 45, 60. And again, we see the very same pictures. The astrocytes shrink. They become smaller and much less complex. So what happens, what we think, is in aging, astrocytes sort of retreat from the synapses, and that impairs, of course, in their ability to buffer potassium, to take up glutamate, and increases the possibility for glutamate to spill over, and that all results in changes in synaptic plasticity. And that is only one example of what happens with astrocytic function. Now, if you look for all other functions, that's what happens in aging. Glutamate homeostasis is down, neurogenesis is decreasing, and neurogenesis, you remember, it's a function of special astrocytes of neurogenic niche. Blood-brain barrier goes down, and astrocytes lose their ability to support blood-brain barrier. Lymphatic clearance is decreased very much, and that is again astrocytic function. Metabolic support goes down in aged brain. Cholesterol synthesis goes down, and without cholesterol we cannot build membranes, of course. And astroglial defense is also impaired in aging. And the same happens with another defensive microglial cell, which is called microglia. In fact, in human brain, about 40% of microglial cells in old age are dystrophic. They lose their functions, they cannot protect. And in a sense, this all decline in astrocytic protection and homeostasis and decline in microglial protection opens the gate for neurodegeneration. So brain aging is not an active destructive process. Brain aging is defined by gradual deterioration of defensive and homeostatic and supportive functions of glia, of all neuroglia. So in a sense, this is probably a target which we have to consider when talking about age-dependent diseases, because this paralysis of neuroglia opens the path to neurodegeneration. Small practical notion: chronic consumption of anti-inflammatory drugs doesn't affect cognitive longevity. Now, in the last 10 minutes, I'm going to talk about atrophy of astrocytes in age-dependent neurodegeneration, and do it from human perspective. And why is that? It's truly important because animals don't have classical neurodegenerative diseases. They don't have Alzheimer's disease. They don't have Parkinson's disease. So we do rely on models. And the models which are currently in use, usually it's overexpression-based mice. So it's mice which are expressing mutant genes which are associated with familial form of Alzheimer's disease, such as amyloid precursor protein or presenilins or tau. And those overexpression-based mice do not recapitulate the disease fully. Actually, they do not recapitulate sporadic disease at all, because it's a very, very different nature. And they do show many, many differences between patients and these model mice. In particular, mice do not have extensive neuronal loss, so their neurons are preserved somehow, they survive. And these overexpression mice, they show quite a bit of this, what we call inflammatory response. So they show quite a bit of reactive phenotypes of microglia and astroglia, although I don't think it's neuroinflammation in earnest. But that is why, because in mouse, with age, number of microglial cells is not decreasing. They don't become dystrophic. And this is one of many, many differences between mouse brain and human brain. And when it comes to astrocytes, that demonstrates you the difference. So this is a rodent astrocyte, and this is human astrocyte. And you see human astrocyte in gray matter is by far larger and by far more complex compared to the rodent astrocyte. So it's very different. And then, as you remember, I mentioned that human brain contains several types of primate-specific glial cells, and in particular, interlaminar astrocytes. So those interlaminar astrocytes, their somata are sitting just under the pia, and then they send very long processes which are penetrating layers. That's why they're called interlaminar. And they usually go down up to layer 4, sometimes even up to layer 5. And now what happens in Alzheimer's disease? This is a healthy male, and this is Jorge Colombo, who found these cells and studied them all his life. This is healthy male. You see there are lots and lots of these very long processes of interlaminar astrocytes. And this is BRAC6 stage. You see all of them disappear. So again, how can we study something which doesn't exist in mouse at all? And that is another example of why this translation between mouse and humans is almost impossible, I would guess. So what happens with different markers of astrocytes in the course of Alzheimer's disease? This is Wendy Noble, my good friend. So what she did, she looked into GFAP expression post-mortem and correlated with different stages of disease. What we see at the beginning, we have an increase in GFAP, and then it's a sudden loss, which corresponds to a change from mild cognitive impairment, of course, to dementia. This is the one. Postmortem of brain banks, again, there are lots and lots of astrocytes in postmortem Alzheimer's disease brain, which are atrophic morphologically, judging either by GFAP or by some other markers. And that is in vivo what happens here. This is Agneta Nordberg, of course, and she followed a patient over the course of the disease using PET scan, positron emission tomography, with deprenyl. Deprenyl is a drug which binds and blocks monoamine oxidase B, which in the brain selectively sits only in astrocytes. And what happens over the course of disease? Mild cognitive impairment, so initial stages of Alzheimer's disease, the signal is increasing. And that somehow could be interpreted that astrocytes are trying to fight against this particular disease. But then we have a switch from mild cognitive impairment to dementia, and the signal falls down. indicating that astrocytes are incapable probably to defend the brain. Stem cell technology, we can take peripheral cells from patients and healthy controls, do whatever, and produce, of course, a culture of astrocytes which look like cultured astrocytes and express all the markers. And now what happens? This is a comparison of healthy control with familial and sporadic Alzheimer's disease. You see severe morphological atrophy. And if you compare neuronal culture with astrocytic culture, again, healthy controls, disease, neuronal cultures look more or less the same. Astrocytic cultures are severely impaired. So in a sense, neurodegenerative diseases like Alzheimer's disease are associated with a massive atrophy of astrocytes which reduces their defensive capabilities. What can we do with that? I don't think it's possible to find a single molecule which will cure, for example, Alzheimer's disease. Most likely Alzheimer's disease, it's an umbrella term which covers many different pathophysiological mechanisms. But what we know, lifestyle changes are very important for our cognitive longevity. Dieting, of course, caloric restriction. What happens with astrocytes in animals which are calorically restricted? So again, this is an astrocyte, this is Scholl analysis, this is complexity, and the same volume fraction. And what happens in caloric restriction? We have more complex astrocyte and more volume fraction. So astrocytes have major presence around synapses, or larger presence, sorry. And that's what it translates into. It translates into increased capacity of removing glutamate. This is glutamate currents, and increased capacity to buffer potassium. And that again translates into increase in long-term potentiation after caloric restriction. And that's how we envisage all that. So caloric restriction sort of more or less makes astrocytic processes growing more, and they have more presence around synapse, better potassium buffering, better glutamate clearance, and better prevention of spillover, which results in increased synaptic plasticity. Environmental stimulation. So we can put animals into physical exercise or enriched environment, and in this situation, this is a triple transgenic animal model, of course. And 9 months of physical exercise or enriched environment actually increases complexity of astrocytes. It rescues the atrophy of astrocytes, which coincides with bettering of their behavioral tests. And the same also applies to neurogenesis. So enriched environment and physical exercise increase neurogenesis in the neurogenic niches. So how to fight brain aging? We have to stay intellectually engaged. We have to maintain physical activity, cardiovascular system, minimize chronic stressors, and maintain a brain-healthy diet. And what is amazing, this recipe was for the first time published about 2,000 years ago by Marcus Tullius Cicero, who said, age has to be fought against. We must combat this aging as we should fight a disease following a fixed regime, taking exercise in moderation, and enough food and drink to strengthen, yet not enough to overburden. And on that, I would like to finish my lecture, and I hope that you will all follow this wisdom of Cicero and have a nice cognitive aging. And these are many people with whom I was working on neuroglial research. Thank you for your attention.